Good afternoon, everyone, and thank you for joining. I'm very excited to be presenting to you all today. My name is Shayan Hobbi, and I'm a healthcare data scientist within the federal practice of IBM. And today I'll be speaking to you all about our efforts to develop a SMART-on-FHIR enabled application focused on the semi-automation of adverse event validation and reporting. So given I only have 10 minutes, this is somewhat of an optimistic agenda, but hopefully we can get through all of it. We'll be covering the current state of adverse event reporting, our efforts to develop the semi-automation solution, challenges and lessons learned, as well as next steps. So starting with the current state of adverse event reporting, the basics of this is essentially that when a medical product goes into market, FDA is in charge of monitoring its safety and effectiveness. In particular, we are working with the Center for Biologics Evaluation and Research within FDA under the Biologics Effectiveness and Safety Initiative, otherwise known as BEST. And BEST goal is essentially to conduct active surveillance for post-market safety and effectiveness of biologic products. And biologic products are essentially anything of human origin that includes vaccines, blood, including blood components and blood-derived products, human tissue, gene therapies, and other advanced therapies like xenotransplantation products. So the current need that we'll be focusing on today is a more robust post-market adverse event reporting system to the FDA. One of the biggest challenges within this space is that these processes are very much manual. Currently, providers, healthcare providers, as well as manufacturers of biologics, are the ones that are mainly reporting to these adverse event systems at FDA. And given that it is manual, and given that there is often not enough data to populate these reports efficiently, FDA is experiencing severe underreporting of adverse events that are occurring with these products. So, as I mentioned earlier, the current state is essentially manual detection of adverse events within EHR systems, and providers often, if they detect anything related to a medical product, they would report it to the manufacturer, and either the provider or the manufacturer report that adverse event to the FDA, or they both do it. So it's very time intensive to manually detect these adverse events. It's often also time intensive to validate the occurrence of that adverse event to make sure a case is actually a case. And it often takes a lot of time to manually report those adverse events using either paper forms or the electronic forms that are available. Often those are just done completely manually. So, needless to say, that is quite a burdensome process that we've been tasked with improving the efficiency of and trying to semi-automate. So our efforts have been focused on semi-automation of this process, and we've developed a FHIR-based solution which is focused on automating the detection of adverse events using AI and other batch detection tools, as well as a SMART-on-FHIR enabled tool, which semi-automates the validation of those adverse events that are detected, and then using that same tool to report the adverse events that have been validated, and populating an ICSR or individual case safety report to the FDA. So this is our current architecture to date. It all starts with getting access to EHR data for enabling detection of adverse events. Currently, we are getting EHR data extracts from several EHR data providers, and that goes through a quality assurance process, particularly to identify cohorts of interest and populations of interest and doing some data quality checks. And then that data is transformed to a FHIR server. And we'll get into this later, but the main reason for doing EHR data extracts and not connecting directly to FHIR endpoints is mainly because of the limitations of the FHIR endpoints currently implemented by providers today. They often lack the data elements needed for biologics related exposures as well as outcomes of interest. So once the EHR data is collected, we have developed several detection models that help detect the adverse event, and then that goes on to the adverse event validation and reporting tool. And if it is validated, a report is auto-populated as much as possible and then reviewed and can be submitted directly to the FDA. So within the tool for the validation step, the tool allows you to view the case definition for the outcome that you are validating. It also allows for assessing the certainty of exposure to a biologic, as well as assessment of causality of the adverse event, as well as essentially identifying all the evidence that may be related to that particular outcome. And then once the selections are made within the tool, as I mentioned earlier, you can semi-automate the population of an adverse event report and then review and submit. So some of the main challenges that we've faced during this development process and learned so far is that often the documentation for the EHR-hosted smart sandboxes, which we tested with Cerner and Epic, are often fairly complex for the provider-facing connections when compared to the patient-facing scope. Although this was relatively minor, we were able to resolve it by contacting the teams who host the smart sandboxes, and we were able to get the provider-facing test connection to work. The other is the biggest challenge was time for providers to review and approve smart app connections to their production FHIR endpoint. This is a significant investment that can take up to 7 to 9 months, depending on the provider, and often using test data and test environments instead of connecting to the provider production systems are not necessarily reflective of real production level data, especially for the types of patients we're interested in, like transfusions. So another big challenge was, one that I mentioned earlier, is the lack of granular data for biologics exposures within the FHIR endpoints hosted by EHRs. So looking at a vaccine adverse event example, we would need these bare minimum of data elements for reporting, which include data elements on the exposure to the product, including NDCs and lot numbers, labs that would rule out other causes of the adverse event, the diagnosis information for the adverse event, as well as any confirmatory information we can get from the unstructured data. And similarly for blood transfusions, one major data element of interest are the ISBT codes. ISBT codes are unique identifiers for both at the product level as well as at the actual blood bag level. And we'll discuss how we're addressing this gap by creating FHIR profiles and implementation guides. So this is a summary of what the main gaps are between US CDI and just going directly to the EHR to get data elements of interest. As I mentioned, NDCs are not included necessarily within US CDI as a must support element, nor are ISBT codes for exposure to blood components. I think we may be running out of time, so I will skip over a lot of these, which essentially highlight the process of how we came up with our implementation guide. But in short, we gathered all the data elements of interest that would be needed for outcomes, exposure, detection, and validation, as well as for actual reporting of the adverse event, and compiled those into FHIR data elements and value sets, and compiled those into profiles and in a larger best implementation guide. And that implementation guide adds in a profile for the biologically derived product, and must support for ISBT codes and NDC codes for getting granular details for exposure to those products. And really the next step here is to work with the proper HL7 work groups to get these data elements added to US CDI for future adoption by providers and EHR vendors. I think I'm out of time here, so we'll move on to questions. And again, thank you to everyone for joining and listening.